Hello and welcome back to our tour of the cell. I have a change of scenery for you. I have a new whiteboard, so I'm hoping that works out well for you. We are officially still into our tour of the cell. We finished a good handful of organelles so far. We started with the nucleus all the way to the peroxisome so far. And I'm hoping what you see with these organelles that we've studied is that they are sort of independent of one another. They're not in contact. There's a lot of cellular signaling going on, but you don't need to worry about that. However, the nucleus is doing what the nucleus does, the mitochondria is doing what it needs to do, it's replicating on its own, doing its own thing. This next set of organelles that we're going to look at, again, organelles, these structures that are functioning inside the cell, these next set of organelles are more like a family, if you will. They're always in contact with each other. They either remain in contact through direct interaction from their membrane, or sometimes part of the organelle literally buds off of the organelle and fuses with another part of another organelle. So it's either by direct communication or indirect communication, all through the lipid membrane. And so this next set of organelles has a special group name to it, if you will. And we like to call these guys the endomembrane system. And there's 5 organelles within the endomembrane system. So let's start there. Big letter E on the last section was peroxisome. So big letter F on here is going to be the endomembrane system. Here we go. The endomembrane system. And so what these are a group of organelles that are either in, they're bound by a phospholipid bilayer like all organelles are inside the cell, but they are in direct or indirect contact through their phospholipid bilayer. So a group of organelles that are in direct or indirect. I know that looks, but that's 2 words. Indirect or indirect. This time it's 1 word. With one another via through their phospholipid bilayer. Okay, now we have 5 different organelles within the endomembrane system. So let's talk about these guys. The first one we've already studied a little bit. And it's technically not an organelle, but it is a structure within the cell that is in direct communication with the next organelle we're about to learn about. So this structure is the nuclear envelope. We learned about this guy already. Now, do you remember what was special about the nuclear envelope? It wasn't just a single phospholipid bilayer, but it was a double phospholipid bilayer. So it had a bilayer and another bilayer. That second bilayer is what's in communication with the next organelle. So what you need to know about the nuclear envelope is that the outer lipid bilayer of the nuclear envelope is in direct communication with an organelle called the endoplasmic reticulum. Now we're going to learn about that guy next, so hang on. But first thing you need to know, nuclear envelope, the outer layer of the nuclear envelope is in direct contact and actually helps make up the membrane of the endoplasmic reticulum. Is in direct contact and actually helps make the bilayer of the endoplasmic reticulum. Okay, I'll draw this out for you in a second too. Let me erase this. We could draw a picture of what this would look like, and then we could get into the endoplasmic reticulum as well. So if this is a typical cell, you have your nucleus. So there's one bilayer. There's my outer bilayer. This organelle that just kind of stems from it outside here. This is my double phospholipid bilayer. Here's my nucleus. And this right here is my endoplasmic reticulum. So what I want you to see is that the outer layer of the double phospholipid membrane actually becomes the membrane for the endoplasmic reticulum. That's why it's part of the endomembrane system. It's in direct communication with this next organelle that we need to learn about, the endoplasmic reticulum. So number 2, the endoplasmic reticulum. It's ER for short, which we're going to use because we like to shorten things. Endoplasmic reticulum, or ER. All right, so the endoplasmic reticulum, the ER, actually is a huge organelle inside the cell. It's a very extensive network of membrane that's just, I drew it very simple here, but if you were to look inside a real cell, you would just see these folds and folds and folds of membrane. And remember, the more folds you have, the more surface area you have. So the more communication can go in and out of the ER, things like that. So this has it's a highly extensive network of membrane within the cell. And inside the ER, whole lots of different functions are going on, which we're going to talk about. But the first thing I want you to know is that little a under ER, it's a highly extensive network of membrane. That's one thing I want you to know about the ER. And these little tubules that get formed here, I drew them very simple. These little tubules that kind of extend here, these little guys are called cisternae. That's a Latin word. So cisternae basically meaning reservoir for liquid. And so the folds are called cisternae. The folds are cisternae. And inside the ER, it's almost like you have another cell within a cell because all of these organelles are wrapped in their own membranes. And inside, you got stuff going on. Inside the ER, the fluid on the inside is called the lumen. So inside the ER, the fluid is called the lumen. Here's where it gets really, really fun about the ER. The ER, actually there's 2 kinds of endoplasmic reticulums. There's a smooth ER and a rough ER. And we've kind of referenced the rough ER in the past. If you remember our discussion about the ribosome, how you have bound and free ribosomes, those bound ribosomes were bound to the membrane of the ER. That's why it's called rough. When you look at it under the microscope, you would see all these little dots on the ER. It looked bumpy and rough. It was because these ribosomes were embedded within the membrane. So we're going to talk about both types. So what I want you to know, let's do this. So there's 2 types of ER, rough and smooth. We're going to actually talk about the smooth first because the rough leads to our next discussion. So let's discuss the smooth ER. Here we go. This will be little I under D. The smooth ER is actually quite fascinating. The smooth ER we literally call it smooth because there's a lack of ribosomes. There's no ribosomes. So we put a lot of nomenclature on the structures within a cell based upon what we used to see in a microscope. And the more detail we got, it was nice to see that actually correlated. It's smooth compared to the rough, just only the lack of ribosomes. The functions of the two endoplasmic reticulums are completely different. And what you need to know is that the smooth and the rough kind of cohabitate together. Usually you'll see the smooth closer to the nucleus, but not necessarily. But the rough, you'll know it's rough because you'll see ribosomes embedded all throughout. So for the smooth ER, it's literally called smooth because there's a lack of ribosomes. Lack of ribosomes in the membrane. But the smooth ER has some highly specialized functions, and it's pretty fascinating how the smooth ER works. The first thing that it does is lipid metabolism. So or lipid synthesis, lipid synthesis. It helps create the lipids we learned about back in Unit 1. So it helps make some oils, phospholipids. It can help with steroids. So depending on the cell type you have, whatever cell you're looking at, if you need more lipids to be made, you're going to have a lot of smooth ER. You're just going to have a bigger portion of smooth ER in there. So the first thing you need to know, this will be a bullet, is it helps with lipid synthesis. Helps make lipids. The other neat thing it does, which I've always found fascinating, is that it helps, there's a couple things, helps metabolize carbohydrates. So the carbohydrates you eat, this helps break them down. So metabolizes carbohydrates. And lastly, this is where it gets really interesting, is that it helps detoxify drugs. And by drugs, I literally mean everything from Tylenol to the naughty stuff that you shouldn't be taking. And so, ever notice how, let's take, unfortunately, alcohol as an example. If someone's drinking alcohol, the first time they drink alcohol, they usually feel the effects of that drug. It's a chemical, so therefore it's a drug, right? But the more and more and more they take it, they don't feel the effects as fast. And that's because everything in our body is adaptable to a certain degree. Your smooth ER can actually grow inside your cells, specifically your liver cells that are helping to detoxify all these things. They can grow and grow and grow. So you actually have to take more of that drug to feel the effects. Same thing goes for painkillers and stuff like that. So you need to be very cautious on how you take care of your body and never abuse any substances because your body can adapt to a certain degree. However, it only adapts so much. And then after a while, you start building up that drug and you get toxicity, and that's bad. So, but taking care of your body and knowing how these things work inside your body is very important. And the good news is, once you stop taking these drugs, your smooth ER can actually go back to a normal size again. So you don't, like, you'll be sensitive again, which is good. So helps detoxify drugs. Helps detoxify drugs. All right, so the other last thing that the smooth ER does, and this is important for your muscle cells, the smooth ER is important for storing calcium ions, specifically in muscle cells. So stores calcium ions. In muscle cells. If you ever get into muscle anatomy and structure, that has a specific name inside the muscle cell. It's not really called the smooth endoplasmic reticulum. It's called the sarcoplasmic reticulum. That's because that area of a muscle cell is called the sarcoplasm, and it's within that area. So but it's essentially a smooth ER. Okay, so that's the smooth ER. Let's do the rough ER now. Here we go. So the rough ER, as you know, is rough because of ribosomes. Rough ER contains ribosomes in the membrane. All right, now if you're thinking ahead, you're probably realizing that the rough ER, obviously it's rough because of the ribosomes, and what is the main function of a ribosome? If you remember, it's to help make protein. So ribosomes make protein, and what's happening is these ribosomes are embedded in the membrane in the ER. They're creating protein, and these proteins actually get made and produced in the lumen of the ER. And what's happening within the lumen of the ER, these proteins, these amino acid chains are being made. They immediately get folded and turned into their proper structure so they can actually become functional. So if you remember back in Unit 1 when we were talking about proteins and amino acids, you would have this long amino acid chain. That would be your polymer of a protein. These amino acid chains have to fold into a primary structure all the way to a quaternary structure, so 4 levels of folding. And within the ER lumen, the surroundings and the chemical ion concentration and everything is just right for those proteins to fold into the right structure they need to be. So, what happens in the rough ER is that proteins made by the ribosomes enter the lumen of the ER and fold into their proper structure. Now that's huge, because if these proteins can't fold into their proper structure, they can't do their proper function. So it's all about structure function. And it depends on the type of protein that you're going to be getting out of this gig, whatever protein it's been coded to create from the ribosome. Sometimes proteins become something called a glycoprotein, which means they get a little bit of sugar attached to that protein because it's like glucose, glyco. It has glycoprotein. That's very typical of proteins that need to be secreted out of the cell. Remember that bound ribosomes create 2 styles of proteins, ones that either function in the membrane or get sent out of the cell. So it just depends on what's happening with this protein. Now this protein, now that gets made, so if this is a ribosome, this protein enters into the ER and then it gets folded into a specific shape. I'll just turn it into a square. Now it's sitting in the ER. Now we're getting ready to tell the story of how a protein, how does it get made, how it got made by the ribosome. We're going to get into those details in another unit. But the protein gets made by the ribosome, it folds up into its proper structure in the ER, and how does that protein get to where it needs to get? Enter the next organelle. So here's what's really, really neat about the ER. Once the protein is folded into its right structure, this protein is actually going to exit the ER and go to the next organelle in line. The next organelle in line is called the Golgi apparatus. So the Golgi apparatus is this organelle that I'll try to draw it all nice and pretty. I'll just make it look like this. This is a cross section of it. So this guy right here is the Golgi. It's called the Golgi apparatus. And what's going to happen is this square protein, I'm just going to erase this for a second, is going to leave the ER. It's actually going to bud off of the ER. It's going to go into, actually I'm going to erase this a bit so I get some more room. It's going to go into a transport vesicle right here, and it's going to enter and fuse with the Golgi apparatus. So this guy, it buds off and fuses with the next one. So for the rough ER, the folded protein, and oftentimes it gets sugars attached to it and stuff like that. The folded protein buds off the ER in a transport vesicle and fuses with the Golgi apparatus. Now we're going to talk about the Golgi apparatus. That's our next organelle line. But that's it for the ER. So once the protein gets made, it's kind of like, okay, I put together this package, now I got to send it somewhere to get shipped. Enter the Golgi apparatus. The Golgi apparatus is kind of like the UPS of our cells. All right, so let's do the Golgi apparatus. This will be the next organelle in our endomembrane system. So we have the nuclear envelope, we have the ER, and now we have the Golgi apparatus. So this will be number 3 in our list. So the way I want you to think about the Golgi apparatus, it's the receiving, let's see, the warehousing, the shipping, or the receiving, if you will, the sorting, and the shipping area of the cell. So this is the receiving, sorting, and shipping protein warehouse. So here's the story of the Golgi apparatus. It's actually quite fun. So we left off in our story of the protein. The protein leaves the endoplasmic reticulum in a transport vesicle. The transport vesicle fuses with the Golgi apparatus, but it only fuses with the specific side of the Golgi apparatus. The side closest to the ER is actually called the cis side. Cis is like same, on the same side. So what happens is the transport vesicle that's carrying this precious protein cargo, the transport vesicle from the ER fuses on the cis side, CIS, cis side of the Golgi. And here's the fun part of how this protein actually matures as it travels through the Golgi. So this protein is now tasked with kind of growing up because it's not completely mature yet. And so what happens, I want you to think of these little stacks of membrane which make up your Golgi apparatus. These are all another stack of membrane. They're also called cisterna as well. What's happening is every time they enter a new pocket of the Golgi, some kind of modification, some kind of tweak happens to this protein to help it grow up a bit. Think of it as you going through kindergarten all the way to 12th grade. What you can do, so if we gave a kindergartner the task a senior in high school should expect to do, that poor kindergartner wouldn't know what to do. There has to be a maturation process of that student so you can actually do a job when you get out of school. And the same exact thing happens to a protein. As it goes through the Golgi apparatus, it gets tweaked and molded and influenced, if you will, by the cell to become what it needs to become. So a modification happens here, and then it's going to butt off again into another transport vesicle. It's going to fuse again here. It's going to get a little modification. It's going to butt off again into another transport vesicle. It's going to butt in. It's going to fuse in again. And every time it's just kind of changing and getting tweaked ever so slightly. And then here, this is its last step. This is basically senior year right here. And what's happening here is that it's basically getting a zip code. It's being told where to go. Imagine if you graduate from college and you want to go to you graduate from high school and you want to go to college, or you want to go take a job somewhere. That you're going to have a new zip code perhaps. You're going to have to be shipped away. And so the same thing happens with the protein. So as it's going from cisterna to cisterna, it's getting matured. Same thing as you go from grade to grade to grade. Before it leaves that last Golgi apparatus cisterna, it receives its molecular zip code, its molecular tag to say, you're a protein that needs to go function in the cell membrane, or you're a protein that needs to get secreted out of the cell to go do its job. Whatever it is, it gets that tag, that zip code, so the transport vesicle that's going to take it where it needs to go knows where to go. These little signals inside the cell are truly amazing and highly specific, and it allows everything to work well. So this whole process of proteins maturing as they go through the cisterna is actually a very functional name. We actually call this cisternal maturation, the cisternal maturation process. So, transport vesicle from the ER fuses on the side of on the cis side of the Golgi. The proteins, let's just write this out. The proteins travel through the cisterna of the Golgi, all the while receiving modifications for function. And the way they travel through, and so travel via transport vesicles. That's really important. So this whole process of them traveling from cisterna to cisterna to cisterna, this is called the cisternal maturation model. The cisternal maturation model. I'm going to put little c over here just so we have it all together. And then finally, before that last transport protein takes the protein away from the Golgi apparatus, the protein receives a molecular tag, a zip code, to give it its final destination place. And the other thing I want you to know is how this side was called the cis side. This side is called the trans side. It's the opposite side. So the final level of protein development, we'll call it that, involves a molecular tag so the protein knows where to go. Knows where to function, I'll say that. And then the very last thing is that the protein leaves the trans side of the Golgi apparatus and a transport vesicle. So protein leaves of Golgi in a transport vesicle. Okay? That is visit for the Golgi apparatus. So now you've seen how the path of a protein